everyone. My name is Julie Chang, and I'm a sixth-year bioengineering PhD student working in the lab of Ovijit Chaudhary at Stanford. My research focuses on studying how mechanics mediate cell invasion. And today I'll give a brief overview of my dissertation work titled Force-Mediated Collective Invasion Through Endogenous Basement Membrane During Breast Cancer. So much of our knowledge and approach to studying cancer has been focused on studying the biology of cancer and focusing on cancer as a genetic disease. However, another approach to studying this complex disease is through mechanobiology, which is mechanics and biology combined. And here we study how cells interact with their physical surroundings. So one analogy to describe mechanobiology at the cellular level are these indoor playgrounds that we might have played on as children. This obstacle course, which we can think of as an extracellular matrix, and the children, which we can think of as cells, must navigate and squeeze through the small spaces in this obstacle course. So in these playgrounds, there's usually a pretty straightforward path where children can navigate from the beginning to the end. So children can crawl through tunnels or slide down slides. However, it would be pretty much impossible for children to squeeze through these mesh nettings in this section of the obstacle course. So the basement membrane is just like this mesh netting shown here. The holes in this netting are a lot smaller than children. And similarly, the pores of the basement membrane are nanometer size, which is too small for cells, which are micron sized to migrate through. So in the basement membrane, the BM is shown here in pink, and it is a meshwork of proteins that form a stiff yet thin barrier that separates the tumor from the extracellular matrix. So normally cells are contained within the basement membrane, but if cells invade through here and escape, they can then enter the bloodstream and spread to the rest of the body. So in this project, I developed a 3D in vitro model of ductal carcinoma to model invasion through the basement membrane. So in this 3D hydrogel system, the cell spheroids, also known as acini, they secrete their own basement membrane, which is here shown in blue. The basement membrane envelops the entire cell cluster. And by using live confocal imaging, we can observe the collective invasion of these epithelial cells through the basement membrane. The hydrogel system used is a mixture of alternate and reconstituted basement membrane. So using this in vitro model, we investigated the roles of protease versus non-protease mediated invasion. So the predominant thinking was that cells would use proteases to chemically degrade the basement membrane. However, more recent studies have shown that cells can use force to navigate their surrounding physical environment. So we used several different protease inhibitors and found that cells still invaded through when there was protease inhibited. And finally, to observe the physical interactions of the cell and the basement membrane, we tagged the basement membrane with a fluorescent antibody shown in blue. So in this time sequence, we see that cells are invading into the IPN at the 16th hour, and the basement membrane is being breached starting at 8 hours. So in these angled views, we can actually see that the basement is being densified and while the cells are invading through. So these are just some of the data that I discussed in my dissertation. So I welcome you to look at my poster to learn a bit more. Thank you.